Tightly bound small molecules add extra functions to proteins. Although the order of amino acids in proteins give these molecules their shape and the versatility to perform different functions, sometimes the amino acids by themselves are not enough. Just as we use tools to enhance and extend the capabilities of our hands, so proteins often employ small, non-protein molecules to perform functions that would be difficult or impossible using amino acids alone. Thus, the photoreceptor protein rhodopsin, which is the purple, light-sensitive pigment made by the rod cells in the retina, detects light by means of a small molecule, retinal, which is attached to the protein by a covalent bond to a lysine side chain. Figure 433a. Retinal changes its shape when it absorbs a photon of light, and this change is amplified by rhodopsin to trigger a cascade of enzymatic reactions that eventually leads to an electrical signal being carried to the brain. Another example of a protein that contains a non-protein portion essential for its function is hemoglobin. See Figure 420. A molecule of hemoglobin carries 4 non-covalently bound heme groups, ring-shaped molecules each with a single central iron atom. Figure 433b. Heme gives hemoglobin and blood its red color. By binding reversibly to dissolved oxygen gas through its iron atom, heme enables hemoglobin to pick up oxygen in the lungs and release it in the tissues. When these small molecules are attached covalently and permanently to their protein, they become an integral part of the protein molecule itself. We will see in Chapter 11 that proteins are often anchored to cell membranes through covalently attached lipid molecules, and membrane proteins exposed on the surface of the cell, as well as proteins secreted outside the cell, are often modified by the covalent addition of sugars and oligosaccharides. Enzymes, too, make use of non-protein molecules. They frequently have a small molecule or metal atom associated with their active site that assists with their catalytic function. Carboxypeptidase, an enzyme that cuts polypeptide chains, carries a tightly bound zinc ion in its active site. During the cleavage of a peptide bond by carboxypeptidase, the zinc ion forms a transient bond with one of the substrate atoms, thereby assisting the hydrolysis reaction. In other enzymes, a small organic molecule serves a similar purpose. Biotin, for example, is found in enzymes that transfer a carboxylase group, COO-, from one molecule to another. See Figure 337. Biotin participates in these reactions by forming a transient covalent bond to the COO- group to be transferred, thereby forming an activated carrier molecule. See Table 3.2, page 109. This small molecule is better suited for this function than any of the amino acids used to make proteins. Because biotin cannot be synthesized by humans, it must be provided by the diet. Thus, biotin is classified as a vitamin. Other vitamins are similarly needed to make small molecules that are essential components of our proteins. Vitamin A, for example, is needed in the diet to make retinal, the light-sensitive part of rhodopsin. How proteins are controlled. So far, we have examined how proteins do their jobs, how binding to other proteins or small molecules allows them to perform their specific functions. But inside the cell, most proteins and enzymes do not work continuously or at full speed. Instead, their activities are regulated in a coordinated fashion so that the cell can maintain itself in an optimal state, generating only those molecules it requires to thrive under the current conditions. By coordinating when and how vigorously proteins function, The cell ensures that it does not deplete its energy reserves by accumulating molecules it does not need, or waste its stockpiles of critical substrates. We now consider how cells control the activity of proteins and enzymes. The regulation of protein activity occurs at many levels. At one level, the cell controls how many molecules of each enzyme it makes by regulating the expression of the gene that encodes that protein, discussed in Chapter 8. At another level, the cell controls enzymatic activities by confining sets of enzymes to particular subcellular compartments, often, but not always, enclosed by distinct membranes, discussed in Chapter 14 and 15. But the most rapid and general process used to adjust reaction rates operates at the level of the enzyme itself. Although proteins can be switched on or switched off by a variety of mechanisms as we see next, all in some way cause the protein to alter its shape and therefore its function. The catalytic activities of enzymes are often regulated by other molecules. All living cells contain thousands of enzymes, many of which are operating at the same time and in the same small volume of the cytosol. By their catalytic action, enzymes generate a complex web of metabolic pathways composed of chains of chemical reactions in which the product of one enzyme becomes the substrate of the next. In this maze of pathways, there are many branch points where different enzymes compete for the same substrate. The system is so complex that elaborate controls are required to regulate when and how rapidly each reaction occurs. The most common type of control occurs when a molecule, other than a substrate, specifically binds to an enzyme at a special regulatory site outside of the active site, altering the rate at which the enzyme converts its substrates to products. In feedback inhibition, an enzyme acting early in a reaction pathway is inhibited by a late product of that pathway. Thus, whenever large quantities of the final product begin to accumulate, the product binds to an earlier enzyme and slows down its catalytic action, limiting further entry of substrates into that reaction pathway. Figure 434. Where pathways branch or intersect, there are usually multiple points of control by different final products, each of which works to regulate its own synthesis. Figure 435. Feedback inhibition can work almost instantaneously and is rapidly reversed when product levels fall. Feedback inhibition is a negative regulation. It prevents an enzyme from acting. Enzymes can also be subject to positive regulation, in which the enzyme's activity is stimulated by a regulatory molecule rather than being shut down. Positive regulation occurs when a product in one branch of the metabolic maze stimulates the activity of an enzyme in another pathway. As one example, the accumulation of ADP activates several enzymes involved in the oxidation of sugar molecules, thereby stimulating the cell to convert more ADP to ATP. Allosteric enzymes have binding sites that influence one another. One feature of feedback inhibition was initially puzzling to those who discovered it. The regulatory molecule often has a shape that is totally different from the shape of the enzyme's preferred substrate. Indeed, when this form of regulation was discovered in the 1960s, it was termed allostery, from the Greek allo, other, and steer, solid or shape. As more was learned about feedback inhibition, researchers realized that many enzymes must have at least two different binding sites on their surface: the active site that recognizes the substrates and one or more sites that recognize regulatory molecules. Furthermore, the substrate and regulatory sites must somehow communicate in a way that allows the catalytic events at the active site to be influenced by the binding of the regulatory molecule at its separate site. The interaction between sites that are located on separate regions of a protein molecule is now known to depend on conformational changes in the protein. Binding at one of the sites causes a shift in the protein structure from one folded shape to a slightly different folded shape. Many enzymes have two conformations that differ in activity, each stabilized by the binding of different ligands. During feedback inhibition, for example, the binding of an inhibitor at one site on the protein causes the protein to shift to a conformation in which its active site, located elsewhere in the protein, becomes less accommodating to the substrate molecule. Figure 436. Many, if not most, protein molecules are allosteric. They can adopt two or more slightly different conformations, and by a shift from one to another, their activity can be regulated. This is true not only for enzymes, but also for many other proteins, including receptors, structural proteins, and motor proteins. The chemistry involved here is extremely simple in concept, because each protein conformation will have somewhat different contours on its surface. The protein's binding sites for ligands will be altered when the protein changes shape. Each ligand will stabilize the conformation that it binds to most strongly, and at high enough concentrations, the ligand will tend to switch the population of proteins to the conformation that it favors. Figure 437. Phosphorylation can control protein activity by triggering a conformational change. Enzymes are not only regulated by the binding of small molecules. Another method commonly used by eukaryotic cells to regulate protein activity involves attaching a phosphate group covalently to one of its amino acid side chains. Because each phosphate group carries two negative charges, the enzyme-catalyzed addition of a phosphate group to a protein can cause a major conformational change by, for example, attracting a cluster of positively charged amino acid side chains. This conformational change can in turn affect the binding of ligands elsewhere on the protein surface, thus altering the protein's activity. Removal of the phosphate group by a second enzyme returns the protein to its original conformation and restores its initial activity. This reversible protein phosphorylation controls the activity of many different types of proteins in eukaryotic cells. In fact, this method is used so extensively that more than 1/3 of the 10,000 or so proteins in a typical mammalian cell appear to be phosphorylated at any one time. The addition and removal of phosphate groups from specific proteins often occurs in response to signals that specify some change in a cell's state. For example, the complicated series of events that takes place as a eukaryotic cell divides is timed in this way, discussed in Chapter 18, and many of the signals generated by hormones and neurotransmitters are carried from the plasma membrane to the nucleus by a cascade of protein phosphorylation events, discussed in Chapter 16. Protein phosphorylation involves the enzyme-catalyzed transfer of the terminal phosphate group of ATP to the hydroxyl group on a serine, threonine, or tyrosine side chain of the protein. This reaction is catalyzed by a protein kinase. The reverse reaction, removal of the phosphate group, or dephosphorylation, is catalyzed by a protein phosphatase. Figure 438a. Phosphorylation can either stimulate protein activity or inhibit it, depending on the protein involved and the site at which it is being phosphorylated. Figure 438b. Cells contain hundreds of different protein kinases, each responsible for phosphorylating a different protein or set of proteins. Cells also contain many different protein phosphatases. Some of these are highly specific and remove phosphate groups from only one or a few proteins, whereas others act on a broad range of proteins. The state of phosphorylation of a protein at any moment in time, and thus its activity, will depend on the relative activities of the protein kinases and phosphatases that act on it. For many proteins, a phosphate group is added to a particular side chain and then removed in a continuous cycle. Phosphorylation cycles of this kind allow proteins to switch rapidly from one state to another. The more rapidly the cycle is turning, the faster the concentration of a phosphorylated protein can change in response to a sudden stimulus that increases its rate of phosphorylation. However, keeping the cycle turning costs energy because one molecule of ATP is hydrolyzed with each turn of the cycle. Gtp binding proteins are also regulated by the cyclic gain and loss of a phosphate group. Eukaryotic cells have a second way to regulate protein activity by phosphate addition and removal. In this case, instead of being enzymatically transferred from ATP to the protein, the phosphate is part of a guanine nucleotide, either guanosine triphosphate, GTP, or guanosine diphosphate, GDP, that is bound tightly to the protein. Such GTP binding proteins are in their active conformations with GTP bound. The protein itself then hydrolyzes this GTP to GDP, releasing a phosphate, and flips to an inactive conformation. As with protein phosphorylation, this process is reversible. The active conformation is regained by dissociation of the GDP, followed by the binding of a fresh molecule of GTP. Figure 439. A large number of related GTP binding proteins function as molecular switches in cells. The dissociation of GDP and its replacement by GTP, which turns the switch on, is often stimulated in response to a signal received by the cell. The GTP binding proteins bind to other proteins to control their activities, and their crucial role in intracellular signaling pathways will be discussed in detail in Chapter 16. Here we shall look at their general mechanism of action by examining the bacterial elongation factor EFTU, a small GTP binding protein that helps to load tRNA molecules onto ribosomes during protein synthesis, discussed in Chapter 7. Analysis of the three-dimensional structure of EFTU has revealed how an allosteric transition triggered by the gain or loss of a phosphate on the bound guanine nucleotide can cause a major shape change in a GTP binding protein. Figure 440 shows how the loss of a single phosphate group, which initially causes only a tiny movement of 0.1 nanometer or so at the binding site, is magnified by the protein to create a movement 50 times larger. Dramatic shape changes of this type also underlie the very large movements created by the types of proteins that we consider next. Nucleotide hydrolysis allows motor proteins to produce large movements in cells. We have seen how conformational changes in proteins play a central part in enzyme regulation and cell signaling. But conformational changes also play another important role in the operation of the cell. They enable proteins whose major function is to move other molecules, the motor proteins, to generate the forces responsible for muscle contraction and many of the dramatic movements of cells. Motor proteins also power smaller scale intracellular movements. They help move chromosomes to opposite ends of the cell during mitosis, discussed in Chapter 18. Move organelles along molecular tracks within the cell, discussed in Chapter 17, and move enzymes along a DNA strand during the synthesis of a new DNA molecule, discussed in Chapter 6. An understanding of how proteins can operate as molecules with moving parts is therefore essential for understanding the molecular basis of cell behavior. How are shape changes in proteins used to generate orderly movements in cells? If, for example, A protein is required to walk along a narrow thread, such as a DNA molecule. It can do this by undergoing a series of conformational changes, as illustrated in Figure 441. However, with nothing to drive these changes in an orderly sequence in one direction only, they will be perfectly reversible and the protein will wander randomly back and forth. To make the series of conformational changes unidirectional and force the entire cycle to proceed in one direction, it is enough to make any one of the steps irreversible. For most proteins that are able to walk in a single direction for long distances, this irreversibility is achieved by coupling one of the conformational changes to the hydrolysis of an ATP molecule bound to the protein, as illustrated in the schematic model in Figure 442. The mechanism is similar to the one that drives allosteric shape changes by GTP hydrolysis. Because a great deal of free energy is released when ATP or GTP is hydrolyzed, it is very unlikely that the nucleotide binding protein will undergo a reverse shape change as required for moving backward, since this would require that it also reverse the ATP hydrolysis by adding a phosphate molecule to ADP to form ATP. Such behavior is thermodynamically unfavorable and therefore highly unlikely. As a consequence, the protein moves rapidly forward. Many motor proteins generate directional movement in this general way, including the muscle motor protein myosin, which runs along actin filaments to generate muscle contraction, discussed in Chapter 17, and the kinesin protein involved in chromosome movement at mitosis, discussed in Chapter 18. Such movements can be rapid. Some of the motor proteins involved in DNA replication propel themselves along a DNA strand at rates as high as 1,000 nucleotides per second. Proteins often form large complexes that function as protein machines. As one progresses from small, single-domain proteins to large proteins formed from many domains, the functions that the proteins can perform become more elaborate. The most impressive tasks, however, are carried out by large protein assemblies formed from many protein molecules. Now that it is possible to reconstruct biological processes in cell-free systems in the laboratory, it is clear that each central process in a cell, such as DNA replication, protein synthesis, vesicle budding, and transmembrane signaling, is catalyzed by a highly coordinated, linked set of 10 or more proteins. In most such protein machines, the hydrolysis of bound nucleoside triphosphates, ATP or GTP, drives an ordered series of conformational changes in some of the individual protein subunits, enabling the ensemble of proteins to move coordinately. In this way, the appropriate enzymes can be moved directly into the positions where they are needed to carry out successive reactions in a series, as, for example, in protein synthesis on a ribosome, discussed in Chapter 7, or in DNA replication, where a large multi-protein complex moves rapidly along the DNA. A simple mechanical analogy is illustrated in Figure 443.